In this lecture, we will discuss the overall scheme which is used in the identification of unknown bacterium. For precise identification of unknown bacterial culture, it is required that we must collect a specific information about that unknown bacterium, and for that various techniques are used. Techniques such as study of various staining reactions of the unknown bacterium, which involves various microscopic techniques, staining such as simple staining, negative staining, which are the basic techniques, and some of the differential stainings like gram staining, acid fast staining, spore staining, capsula staining, and flagella staining. So the staining reactions of the unknown pathogen, it will help us get a specific information such as the basic cell shape, the cellular arrangement of cells, the gram nature, whether the cells are acid fast, whether the cells are sporulating, whether they are capsulated, and if they are flagellated, then what type of flagellar arrangement is present on the cells. This information, it can only be obtained by recording various staining reactions of the unknown bacteria. The identification can also be done by recording physiological properties of the unknown bacterium, that is the temperature required for the optimum growth, oxygen tolerance of that unknown bacterium, the pH required for the optimum growth, some other peculiar requirements such as requirement of light, requirement of salt concentration, etc., etc. So these physiological properties or physiological needs, they will help us identify which type of bacterium the unknown bacterium is. For example, if the bacteria grows optimally in between 20 to 40 degree, then you can consider it as a mesophile. If the cells, they do not require any oxygen, then they can be considered as a stringent anaerobe. If the pH required for the optimum growth is 6.6 to 7.4, then the cells can be considered as a neutrophiles. If the cells require high salt concentration, the salt concentration as high as 5%, then they are halophiles. From this information, it is possible to identify the unknown bacterium. Sometimes identification is done easily by recording the cultural characteristics of the unknown bacterium. It involves study of colony characteristics over solid agar media, study of growth patterns in the liquid culture media, and a recording of gelatin liquefaction patterns of the unknown bacterium. From the colony characteristics, it is easy for identification. The preliminary identification can be done easily by recording the solid growth patterns as well as the liquid culture growth patterns of the unknown bacteria. Gelatin liquefaction patterns can also help in identifying the bacterium. And most importantly, biochemical reactions of the unknown bacterium. So variety of different reactions or biochemical tests are there. These tests, based on the results of these tests, we acquire specific information such as whether the cells are fermenting the sugar, whether they are producing acid or gas, whether the cells are aerobic, whether they are positive for oxidase test, whether the cells are positive for catalase enzyme, whether the cells are capable of reducing nitrates, the tests like urease test, and some of the important tests like MVIC tests, results of MVIC tests. It is not possible to discuss all the biochemical tests here, but we will discuss some of the significant tests in this lecture. Simple staining, so this is a very simple technique which utilizes only one type of basic stain, and this help in identifying the cell morphology. So this is the procedure of cell simple staining. A smear of the bacterial suspension or bacterial culture is prepared. The smear is air dried, then it is heat fixed, and after staining, we wash the slide, we air dry the slide, and we observe it under the microscope. If the cells appear just like this, it is a staphylococcus culture. The morphology can be identified as the staphylococci. Negative staining, an acidic stain such as nigrosin, India ink, rose Bengal is used for the test. Here no heating is done so that the heat sensitive components of the cell remains in their normal form. So the cell 1 to 2 drops of bacterial culture is taken near the edge of the slide with the help of inoculating loop. 1 to 2 drops of nigrosin is added to the bacterial culture. Both the drops are mixed and the mixture is spread with the help of another slide. This is preparation of direct smear. After the smear is air dried, it is directly observed under the microscope. We see the colorless cells and the colorful background. Gram staining, so it is important differential staining technique which helps differentiate gram positive and gram negative cells. So first the cells are stained with crystal violet. It takes nearly 1 minute. After that the slide is washed. Then the grams iodine, which is a mordant solution. Mordant is a stain fixative. It is added on the slide. After the treatment with grams iodine, after 1 minute the slide is rinsed again. Then the slide is decolorized. For decolorization we use 95% ethanol. The decolorization steps lasts hardly for 30 seconds. Thereafter we wash the slide again and then we counter stain it with some counter stain like safranin or basic fuchsia. And after sufficient reaction time we wash the slide again, and thereafter the slide is dried and then it is observed under the microscope. If we see the purple colored cells, that is retenance of primary stain, then the gram nature of the bacterium is gram positive. If we see the pink cells, that is retenance of counter stain, the cells are considered as gram negative. Acid fast staining is another important differential staining. It is ideal for the identification of acid fast bacteria. The Mycobacterium tuberculosis, causative agent of TB, it can be easily identified from the clinical samples by doing acid fast staining. The procedure of acid fast staining is like this. Smear of bacterial culture is prepared. The smear is covered with the filter paper strip. A carbol-fuchsin stain is added to that filter paper, and that slide is heated over the hot plate or sometimes over the boiling water. After 5 minutes of heating, the paper strip is removed and the slide is washed for 30 seconds. Thereafter the decolorization is done. For decolorization, a strong decolorizer, that is acid alcohol, is used. After 10 to 30 seconds of decolorization, the slide is washed again, and thereafter the counter staining is done. For counter staining, methylene blue reagent is used. Methylene blue treatment lasts only for 2 minutes. Thereafter the slide is washed again, and then the slide is dried or it is blotted with some blotting paper and observed under the microscope. If we see the pink colored cells, then they are then these cells are acid fast bacilli. If we see the blue colored cells, then these cells are non-acid fast. The cells, that is rod shaped cells in a chain, that is streptobacilli, appear just like this after simple staining. Staphylococci appear like irregular clusters of spherical cells or cocci. against the colorless background. Endospores of the bacteria appear just like this. So this is after the superfilter staining. We see the green colored spores and pink colored vegetative cells. After Dorner's technique of endospore staining, we see the colorless rod shaped cells containing pink colored endospores, and this is the diplobacilli, rod shaped diplobacilli are the cells, colorless cells after negative staining. The difference in the gram positive and the gram negative cells is visible through the diagrams. This is the procedure of acid fast staining. The oxygen tolerance can be recorded by observing the liquid growth patterns. If surface pellicle is visible, then the cells are stringent aerobe. If the bottom sediment is visible, then the cells are strict anaerobe. If uniform growth is present throughout the depth of the culture, then the cells are facultative anaerobe or aerotolerant anaerobe. And if the growth is present below the surface but above the center, then they are microaerophile. Similarly, the other requirements like pH and temperature are recorded. The cultural characteristics include various growth patterns of the bacteria over the solid culture media. So these are some of the growth patterns which are shown in the diagram. The colony characteristics involve the shape of the colony, size of the colony, margin of the colony, elevation, texture, appearance, pigmentation, optical property or opacity, and consistency. Sometimes the characteristic odor is also recorded. So if the cells are E. coli, the cells appear like the spherical colonies. The colonies appear pink on the McConkey's agar. The colonies have the convex elevation, regular margin, and the colonies are opaque with moist consistency. Over mannitol salt agar, the bacteria like Staphylococci appear just like this. So this is the colony characteristics of Staphylococcus aureus. The yellow colored colonies surrounded by the yellow halo are clearly visible. The bacilli, they are visible as a large flat colonies on the nutrient agar with irregular margin or lobate margin, filamentous colonies. The gelatin liquefaction patterns of the bacteria can also be recorded as a means of identification. So these are various types of gelatin liquefaction pattern. Gelatinase enzyme which is present with the bacterium, it is responsible for the liquefaction pattern in the gelatin medium, which is the solid medium. So here we see different types of liquefaction pattern, and from these liquefaction patterns it is possible to identify the bacterium. The biochemical tests are very important, and some of the significant tests include sugar fermentation test with and without acid or gas production. So here after the sugar fermentation test we see the color change in the medium. This is the positive acid production, and sometimes if the bacteria are producing the gas, then there is a formation of bubble in the medium. The other important tests include MVIC tests. So this is the positive test result for each and every test. This is a positive indole test. This is positive methyl red test. So indole production test and methyl red test, they are positive if the bacterium is suspected to be E. coli. It can be confirmed from these two test results. In case of E. coli, the Voges-Prosker test and citrate utilization tests are negative. If we get these type of results, we can easily identify the bacterium as E. coli. Indole production test, it is done with the help of peptone water as a substrate, or indole motylate medium or SIM medium is used as a medium. Methyl red test and Voges-Prosker test, it is done with the help of glucose phosphate broth as a substrate medium. and citrate utilization test is done with the help of Simon citrate agar. So the procedure for the test include inoculation of the test bacteria into a variety of different media. So these four media are used. After inoculation, the media tubes are incubated, and after the incubation, we see the results. So after addition of covax reagent, if red coloration is there at the top, it indicates production of indole. Amino acid tryptophan, it is broken down into indole, pyruvic acid and ammonia. Pyruvic acid and ammonia are utilized by the cells while indole accumulates as a byproduct. This indole reacts with the covax reagent, which is a type of aldehyde, forming a cherry red colored compound called rosindole. The cherry red coloration, it indicates positive indole production. Methyl red test. So methyl red is a pH indicator. At a pH 4 and below, it is red in color. The cells like mixed acid fermenters, for example E. coli, produce variety of different acids, overcoming the buffering capacity of phosphate buffer and reducing the pH below 4, and that leads to change in the color of the pH indicator. After incubation of that media, that is glucose phosphate broth, when the methyl red indicator is added, and if there is a red coloration, then it is considered as a positive acid production, and the bacteria is said to be positive for methyl red test. This is the negative test result, which is in case of the butanediol fermenters. Bogus Prosker test, it confirms the presence of butanediol fermenters. The glucose is converted to butanediol, and during this conversion, acetone, that is acetyl methyl carbinol, it is produced as a byproduct, as it is produced as a intermediate. This acetone, it reacts with alpha naphthol in presence of KOH and alcohol, and there is a formation of diacetyl. The acetone is oxidized to diacetyl, and this diacetyl reacts with creatinine or guanidine component of peptone, forming red coloration. So positive red coloration after addition of Barrett's test reagent is a positive test result in case of bogus Prosker test. And the last test is citrate utilization test. The ability of bacterium to utilize sodium citrate and to grow in the medium can be recorded by sodium citrate utilization test. So Simon citrate agar is inoculated with the test bacterium. After the utilization of citric acid, there is a production of pyruvic acid, oxaloacetic acid and CO2. The CO2 reacts with the excess sodium which is present in the medium, and there is a formation of sodium carbonate. The sodium carbonate is an alkaline end product. It increases the pH of the medium, and increase in the pH of the medium can be indicated by the blue coloration in the slant, as the indicator bromophenol blue changes the color from green to blue. The bacteria like Klebsiella pneumoniae or Enterobacter aerugine, they show these two tests positive, that is bogus Prosker test and citrate utilization test. Bogus Prosker test is positive because the cells, they produce acetone as an intermediate for 2,3-butanediol, and the cells are capable of utilizing the citrate as the only source of carbon which is present in the Simon citrate agar. If we see these test results, that is indole test negative, methyl red negative, bogus Prosker positive, citrate utilization positive, then you can confirm that the bacterium is Klebsiella pneumoniae. This is another important test, catalase test. So the pathogenic variety like Staphylococci, it is tested by the catalase test. H2O2, which is hydrogen peroxide, it is broken down by the catalase enzyme into H2O and oxygen, and the oxygen, it comes out in the form of effervescence or bubbles. So the test is done either by directly pouring the hydrogen peroxide over the slant, or the bacteria, they are added on the slide in the drop of hydrogen peroxide, and the effervescence of oxygen are recorded as a positive sign for the catalase enzyme or catalase test. Nitrate reduction test. Some bacteria they are capable of reducing the nitrates into nitrites, and this reduction it is tested by nitrate reduction test. Oxidase test, it is another important test. All aerobic cells they are tested for oxidase test. If the cells are positive for oxidase enzyme, then they will produce the purple color. The oxidase reagent it reacts with the bacteria, and there is a formation of purple color on the paper strip loaded with the oxidase test reagent. So these are some of the important tests. So by recording all these tests, by recording the results of all these tests, we can identify the unknown pathogens.